Good morning to all. Today we are going to start the microbial metabolic pathways, and in this topic we will see how different microorganisms they are producing their energy, and though that energy they will utilize for various purpose like transporting the molecule, producing molecule, and in bigger term you can say that they are utilizing that energy in the growth and development and reproduction. We saw catabolic and anabolic reactions in the microorganism, and these reaction is happening in the microorganism in large scale. In the cytoplasm, it is happening in the mitochondria, and in the nucleus also it is happening. So catabolism and anabolism, that these are all two interrelated terms, and the breakdown of the complex molecules. When any complex molecule in catabolic and anabolic reaction, what is the major difference? So we already saw the differences between these two terminology. The catabolic reaction or catabolism, where the larger complex molecules they are breaking down into the simple molecule. So when they are breaking down into the simple type of molecule, their energy will be released, and that energy they will utilize for growth and the development. So in catabolism or catabolic reaction, what is happening? The large type of molecule is breaking down. So in between the large type of molecule, many bonds will be there, and those bonds when it will be broke, they will release the energy. Okay, so energy is a main thing which is required for the any organism to grow and develop and to do reproduction. So organic and inorganic reactions going on in the uh, organism, and that complex organic molecule forms in simpler type of molecules, and we are calling anabolic reactions. So catabolic reactions and anabolic reactions is happening. See, one example I want to give you, because we all know how the simple molecule like glucose, which is converting into the waste product like acids, carbon dioxide, and they will release in the outside the cell. And in between, when this complex molecule is processing, they are producing the energy. So the energy can be utilized for the cell wall, membrane, ribosome and surface structure maintenance. Then that energy also can be maintained for the protein production, nucleic acid replication, and this energy can also utilize for the amino acid production, and later on this amino acid they will utilize for the protein and nucleic acid production, and protein and nucleic acid can be utilized for the other type of organelle production, and these organelles is the part of the cellular machinery. Okay, so the energy from the catabolic and anabolic reactions, it will be utilized for the growth and the development. So these two things is interrelated, and the energy through the ATP cell is utilizing. Now, various terminology we are going to use in this presentation. We will first see what terms is there and how it is defined. So enzymes. So enzymes is a biological catalyst, and it is present in every type of organism. Some organism they are having some different type of enzymes. Some organism they are having other type of enzymes, and their main function is catalyzing the reactions. And when they are catalyzing the reaction, so the substrate can able to convert into the product. Okay, so what they are doing, they are lowering the activation energy so that substrate can easily convert into the product, and maybe energy will release, or maybe some reaction required energy also. So various type of reactions is there, but the enzymes are different types, and they are doing catalytic activity. So enzyme nature also proteinaceous. They are all synthesizing from transcription and translation. So after the translation, they will be modified and they will do their function in the cytoplasm and in the many type of cellular organelles. Okay, then second type of biomolecule is adenosine triphosphate, or in short form we are calling ATP. In ATP, if you will see, adenine is there and three phosphate molecules is attached to the adenine. Okay, so it serves as a energy molecule, and when a phosphate molecule is released from the adenosine triphosphate, so the high energy bond will be broke down and it will release the energy. Okay, so how it is happening when a ATP molecule is there in the cytoplasm, this ATP is having three phosphate attached to the adenosine. So when a phosphate will be released from the adenosine triphosphate, so the bond between the adenosine and last molecule, third molecule of the phosphate, it will break down. When it will break down, what will happen, the energy will release because the phosphate molecules, it is attached with the adenosine with the high energy. So adenosine triphosphate is a high energy molecules, and when the phosphate is releasing, it will produce the energy, and it will convert into adenosine diphosphate. Then energy source, so compound that is oxidized to release energy, also called an electron donor, and they are the main energy source. So like ATP is an energy source because when phosphate is releasing from adenosine, it will release the energy also. And when the adenosine diphosphate convert into or change into adenosine triphosphate, it required one phosphate molecule and it required energy to form a bond. So ATP, it is a cyclic reaction because ATP can able to transport a adenosine diphosphate, and adenosine diphosphate also can convert into adenosine triphosphate. So it is a cyclic type of reaction. Then electron carrier. See some molecules in the cytoplasm. They are carrying the electrons from one molecule to another molecules. Okay, so like they are example like NAD plus, NADP plus, FAD, and NADH, NADPH, and FADH2. These type of molecules is there. They are basically what they are doing. They are carrying the electrons from one point to another point. So reactions is going on all over the cytoplasm, but the electron carriers are the same. These are all the molecules are electron carriers. Then precursor metabolites. These precursor metabolites that links the anabolic and catabolic pathways. And what they will do, they will provide the substrate for the enzyme, and they will utilize to form a product. Okay, so at the end, the product will be formed from the precursor molecules only. So precursor metabolites or precursor molecules is there, and the precursor molecules will be catalyzed by the enzymes, and then it will be converted into the product. So we have pyruvate, acetyl coenzyme A, glucose 6-phosphate is there. So many type of precursor molecules is present in the uh, cytoplasm. Now enzymes. So there is no one type of two type of enzymes is there. We know various type of enzyme families is there, and in that families many type of subgroups is there. So we will discuss those all type of very important enzyme for the metabolic and anabolic pathways. So for some enzyme is there, oxidoreductase enzyme will do these oxidoreductase enzyme oxidation and reduction reaction they will do. So one example is there, lactate dehydrogenase. This lactate dehydrogenase is required to convert into pyruvate and NADH and hydrogen ions to lactate and NAD plus. So lactate dehydrogenase convert pyruvate into the lactate, and it will side product NAD plus will be produced. This NAD plus is a electron carrier. Okay, then second example is transferase enzyme. This transferase, see by the name itself you can understand what they are doing. They are transferring. That's why name is transferase. There is the they are doing oxidoreductase. So oxidation and reduction reaction is going on. So transferase enzyme, what it will do, it will involve the transfer of the group between the molecule. So reaction involving the transfer of the various type of group, they will transfer from one molecule to another molecule. So one example is there, aspartate carboyl transferase. This aspartate carboyl transferase, what it will do, it will functional on the aspartate and carboyl phosphate convert into carboyl aspartate and phosphate. Okay, so this reaction is catalyzed by the aspartate carboyl transferase. Then hydrolase. So hydrolysis reaction they are doing. That's why hydrolase their name it is. Okay, what the example is the glucose 6 phosphatase is there, and this glucose 6 phosphatase is functional on the glucose 6 phosphate. It will react with the water and then it will convert into the glucose, and one phosphate molecule which is part of the uh, glucose 6 phosphate it will release. So phosphate molecules is attached on the sixth position of the glucose. So this is one molecule of glucose is attached on the sixth position of the glucose. That's why this glucose 6 phosphate. When this glucose 6 phosphate react with the water, it will release the phosphate molecule from the glucose 6 phosphate and it will convert into the glucose. For this reaction, we need glucose 6 phosphatase enzyme. Okay, it is a type of hydrolase. Then lyase. Lyase removal of the group from the double bond or addition of the group from the double bond. So what this enzyme will do, it will release or it will add the specific group on the double bond. So double bond, if it is there in the any molecule, it will release or it will add the double any functional molecule. So example is Cl malate is there and it will convert into fumarate and water molecules will be released. And we required fumarate hydratase. Okay, so fumarate hydratase is required to convert into L-malate to fumarate and water will be released. Then isomerase enzyme is there. It will what it will do? It will react involving in isomerization. So isomerase type of enzymes, what they will do? They will be involved in the isomerization of the molecule. Now L-aniline, see isomerase L and D form, two forms it will be there in the isoforms. So L form will convert into the D form. So L form is there, mirror image, and it will convert into the D-alanine. So L-alanine is converting into the D-alanine. This alanine is an amino acid. See, L type of alanine is converting into the D type of alanine. They are the isoforms, and isoforms is converting into the alanine racemase enzyme because these are the racemose type of molecules, mixture of these molecules. This orientation only different, but molecular weight is same. Okay, so they will convert into the L to D form. Okay, and side by side D form also can able to again convert into the A form. So alanine racemase is required to change into the isomerization. Then ligase is there. So ligase is basic enzyme which can able to join two molecules using ATP energy. See, when the DNA is having gap in between, and these DNA gaps can be filled by the ligase enzyme. So they are joining two molecules and it requires the energy. When DNA is having gap, that gaps will be filled by the enzyme. It required a catalyst, and these ligase can able to fill the gap. So one example is there, glutamine synthetase. This glutamine synthetase, what it will do, it will functional glutamate and ammonia, and it required ATP. Then it will convert into the glutamine, which is a amino acid, and ATP convert into the ADP, and one phosphate molecules will be released. Now we know that there is a three type of energy source. First one, the autotroph is there, the microorganisms which are getting energy from the sunlight. They will capture the sunlight, and for capturing the sunlight, they are having specific type of molecule like chlorophyll is there. Okay, chlorophyll A, chlorophyll B is there in the chloroplast, and they will this photosynthetic molecule can able to capture the sunlight, and it will through the chemical reaction it will produce the energy. This energy is captured by the molecule and it will save in the cytoplasm, and this energy will be used for the growth and the development. So this is a one way we are calling autotroph. So plants and autotrophic bacteria, they are producing this type, and photoautotroph especially. We are talking about the photoautotroph. So the organism which is utilizing sunlight as an energy source and converting into the work in between the energy they will utilize. Then next is chemoorganotrophy. The chemoorganotroph organism is there. They will utilize organic compounds and they will oxidize organic compound to produce the chemical energy. And this chemical energy later on will be utilized for the growth and the development. Then chemolithotrophs also there, and they are utilizing inorganic type of compounds. These inorganic type of compounds can be oxidized, and they will again convert into the simple type of inorganic compounds, and in between they will release the energy. This energy can be stored in the cytoplasm, and whenever they require they can do work, and they will do the growth and development and reproduction type of work. Okay, so three type of sources there. First one is the sunlight, second one is the organic compound degradation or reduction, or inorganic compound reduction. So organic and inorganic compound reaction is there, and they are capturing the sunlight from the sun. Whatever energy they are getting from the sun, they will capture. So these three sources is there. Then role of ATP. See, this is the structure of ATP. Here you can see it is adenosine molecule is there. One pentose for pentose sugar is there, and through pentose sugar, one adenosine is attached, and three phosphate molecule is there. This is number one phosphate, number two phosphate, and number three phosphate molecule. So when this molecule, this phosphate molecule released, so what will happen in between? One bond is there, and this high energy bond will be breakdown. When this high energy bond will be breakdown, so the energy will be released. So this triphosphate convert into the diphosphate. So diphosphate also can able to release one more phosphate, and it will convert to adenosine monophosphate AMP. So AMP can able to again convert into ADP by absorbing the energy, and phosphate molecule can able to attach again, and it will convert into the AMP into the ADP adenosine diphosphate. And again one more phosphate molecule, when it will attach, it will adenosine diphosphate will convert into the adenosine triphosphate. So ATP, AMP or ADP, they are all related with the cyclic type of reaction, and they can join and form a ATP. Okay, so basically if you will see, the ATP is the energy currency of the cell, and it is producing the energy, and whenever the excess amount of energy is there, they will make the microorganism and other type of cells, they will able to make the ATPs, and they can store this ATP, and whenever they require the energy, this phosphate molecules will be released from the ATP, and they will able to produce the energy. So energy is released and whenever they are required, and this phosphate bond will be broken down, and the in between after the breaking, the energy will be released. Okay. Now, how the ATP is producing? So, bacteria use many type of mechanisms for the production of ATP, and basically, when they are having energy, they will convert ADP into the ATP. Okay, so here the one example is there. We have glucose to pyruvate conversion, and this glucose will convert into the cyclic method, and ATP will convert into the ADP, and here also ATP will convert into the ADP. But when aldolase is converting into the glyceraldehyde, and then one this phosphate phosphoglyceraldehyde glycerate is there, and they are converting into the 3-phosphoglycerate, it will produce the ATPs. Okay, then again here also phosphophenopyruvate is there, and it is converting into pyruvate, and this is also generating a ATP. So ADP will convert into the ATP. So substrate here it is a glucose, and the final product will be the pyruvate. And in between the many reactions is going on in the cytoplasm, and through this reaction ATP will generate. Now, so oxidative phosphorylation also is happening in the microbial cytoplasm, and through this oxidative phosphorylation, ATP generation is happening during the electron transport chain. We will discuss in the later slide these electron transport chain. Then photophosphorylation also there, and it is happening only in the phototrophs. So the organism which is doing the or which is capturing the sunlight, they are having photophosphorylation, and they will drive the ATP using the radiant energy of the sun. Sun is producing the high energy as a photon particle. These photon particle will be captured by the uh, pigment pigment molecule present in the chloroplast, and that pigment molecule of the chloroplast, A, B, carotenoid, rhodopsins, they will capture this photon, and they will convert this photon energy into the ATP, and ATP will be saved in the cytoplasm, and whenever they require the energy, they will produce. Okay, now metabolic pathways for the energy production. So various pathways is there. We saw glycolysis pathway already, where the glucose is converted into the pyruvate, and in between so many compounds is there, or reactions is happening, and many other enzymes also required to complete. Each step of this reaction is required an enzyme, and it will require also energy. And in between, when three carbon stages is there, three carbon molecule is reacting with other, uh, they are the substrate, they are reacting and converting into the product. They required energy here in the 6 carbon stage, and in 3 carbon stage they will form the energy as a ATP. So it is a, we, in other terms, embedded Merov Parnes pathway we are calling, and it's happening in the cytosol. So cytoplasm is the main part where the this glycolysis is happening. So aerobic and anaerobic type of bacteria, what they will do, they will require the oxygen, and they both have aerobic and anaerobic this type of glycolysis pathway. So three phases is there. Preparatory phase is there, where the cell required the all type of enzyme to finish this glycolysis, and the substrate. See, glucose is required. Without glucose and enzyme, it will this reaction will not be complete. So preparatory part phase is there, where the uh, substrate and enzyme will be prepared and will be ready for the glycolytic pathway. Then splitting phase is there, where the glucose is split into the 6 carbon. See, glucose is a 6 carbon molecule. So it is converting into the 3 carbon molecule. Okay, so it is reducing. Okay, so energy generation phase will be there in the 3 carbon phase. See, ADP is converting into the ATP in the 3 carbon phase. So two molecule of pyruvic acid will be synthesized in the each glucose. For one glucose reaction in the glycolytic pathway, two molecule of pyruvate or pyruvic acid will be produced. And net gain, if you will see in the glycolysis, two ATP will be generated. So NADP, NADH6 ATP also will be produced ultimately, and if you will see net gain, it is 2 ATP in the glycolysis. So alternative of the glycophase. Many bacteria they have another pathway in addition to glycolysis. They can do glycolysis also, but in alternative pathways also they are doing to produce the energy. And these two alternative pathways, pentose phosphate pathway and Entner-Doudoroff pathway. So in pentose phosphate pathway, what is there? Hexose monophosphate shunt also we are calling pentose phosphate pathway, and it occurs simultaneously with the glycolysis and provide breakdown of both pentose sugar and glucose. So it is another pathway where the pentose sugar or glucose is breaking down and they are utilizing to produce the energy. Okay, so what important feature is there? It is a intermediate pentoses are used for nucleic acid synthesis, amino acid synthesis, and glucose from the CO2 in photosynthesis. So intermediate various purpose they are synthesizing, like nucleic acid it will synthesize, amino acid also synthesizing, then glucose molecules it will utilize to form the carbon dioxide, and this carbon dioxide will be released in the environment. Then it is very important reaction, since it important because it is produce the it will reduce the coenzyme NADPH. Okay. Then second important pathway is Entner-Doudoroff pathway. So Entner-Doudoroff pathway also utilize glucose and it will convert into the pyruvate. Okay, so bacteria having enzyme from the Entner-Doudoroff pathway can metabolize without the glycolytic pathway. So this is the pathway where the initial substrate initial substrate is glucose and product will be same the pyruvate, and it is happening in the some gram negative bacteria like Pseudomonas species, Rhizodium species, and they are not generally found in the gram positive bacteria. So only gram negative bacteria they are doing this Entner-Doudoroff pathway. And the ultimately if the bacteria is following Entner-Doudoroff pathway, it will produce one molecule of NADH, one molecule of NADPH, and one molecule of ATP form one molecule of glucose, and the ultimately two molecule of pyruvate will be synthesized. Okay, then cellular respiration and fermentation also happening in the cell, and this cellular respiration and fermentation happening in the pyruvate obtained from the when the glycolysis is over. So after the glycolysis will be two molecule of pyruvate will be obtained from the glucose breakdown. So in the glycolysis, ultimately we will get the two molecule of pyruvate. And in the respiration, what is there? The ATP generating process and the oxidative oxygen is required in, then we are calling oxygenic reaction, means oxygen is required, and final electron acceptor is in an inorganic molecule. So type of respiration is there, aerobic and anaerobic, where the if oxygen is required as a electron acceptor, so it is a aerobic respiration. Okay, and basically those organisms which is doing growth and development in the presence of oxygen, they are the aerobic respiration. They are doing only aerobic respiration, and some microbes they are doing anaerobic respiration. So their final electron acceptor is inorganic molecule. They the oxygen is not required and they can grow without oxygen. So the final electron acceptor is a inorganic molecule, not oxygen. So that's why they can able to grow and they can able to develop in the anaerobic condition, because they can able to do anaerobic respiration. So very good example is there when we are producing alcohol through fermentation of Krebs and sugar molecules. So this reaction is catalyzed by the microorganism. Microorganisms produce the anaerobic respiration important enzymes, and the final product will be the alcohol. And in between this glucose molecule or fructose and basic molecule of the sugar can be able to convert into the alcohol. So anaerobic respiration is required where through the aerobic reaction the glucose will convert into the alcohol. Now, Krebs cycle or TCA tricarboxylic acid cycle. So, Krebs cycle is happening in the second phase of the aerobic respiration, and it is happening in the cytoplasmic membrane, not in the cytoplasm. And pyruvate formed in it enter into the Krebs cycle. So, pyruvate we receive from the glycolysis pathway, and there are two molecule of glycolysis pathway which came from the one molecules of the glucose. Then these pyruvate forms enter into the TCA cycle. So that same molecule of the two pyruvate, it is the basic molecule, or you can say starter molecule or substrate for the Krebs cycle. Okay, and it is converted into the acetyl coenzyme A. And many in between many reactions is happening. So start with the condensation of acetyl coenzyme A and oxaloacetate. So acetyl coenzyme A is oxidized and released as a carbon dioxide, and oxaloacetate is regenerated. So here is the Krebs cycle in detail, and we'll see how it is happening. See, it is a cyclic reaction. So in a cycle, the initial acetyl coenzyme A is required, and it will react with, it will be able to produce citrate, then acetonitrate, isocitrate, alpha ketoglutarate, then it will convert into succinyl coenzyme A. Succinyl coenzyme A, it will able to produce guanine diphosphate 2 guanine triphosphate, which is a low energy molecule. Then succinyl coenzyme may convert into the succinate, and this succinate will convert into the fumarate. Okay, then fumarate will convert into the malate, and malate will convert into oxaloacetate. Then again oxaloacetate will able to react, and it will produce citrate. So again the cycle will go on like that. And these are all the different type of carbon based molecule. See, this is a six membered carbon molecule. The carbon chain 6 is there, and if you will see, this is a 5 carbon is there in the ketoglutarate, and succinyl coenzyme A, succinate, fumarate, malate, and oxaloacetate. It is a 4 carbon molecule. Acetyl coenzyme is a 2 molecule of carbon. So pyruvate is the basic molecule or basic substrate which is required to start the Krebs cycle, and it is coming from the glycolysis. So here how it is happening, and side by side we have energy production steps, and carbon dioxide, it will be produced through Krebs cycle, and it will release in the environment. Okay, so if you see the TCA cycle, 3 molecule of NADH, 1 molecule FADH2, and 1 GTP molecules will be produced. Okay, so all the NADH and FADH2 enter into the electron transport chain, and finally ATP will be generated. So NADH and FADH2, we are calling 3 NADH and 1 FADH2 will be produced through Krebs cycle. And after this 3 NADH and 1 FADH will travel to the electron transport chain, where it will able to produce the ATPs. So how it is happening? So electron transport chain is can able to produce ATP using the NADH and FDP. So it consists of a sequences of the carrier molecules through which electron will be passed in the various plasma membrane, having the specific type of enzymes, and these enzymes can able to convert the NADH2 and FADH2 into the ATP. Okay, so reduction and oxidation is happening in the plasma membrane. So some molecules is required in the plasma membrane, like flavoprotein is required in the plasma membrane. This flavoprotein, what it will do, it is a coenzyme derived from the vitamin B2 and perform alternating oxidation and reduction reaction. And ubiquinones also there, and it will able to produce small non-protein carriers. And cytochrome also required, and what it will do, it is a protein with iron containing group, and heme type of group means iron Fe type of. So here is the electron transport chain. How in the electron transport chain, if you see the NADH which is coming from the Krebs cycle, it will capture by the various enzyme present in these very small proteinaceous molecule present in the plasma membrane, and through the successive carbon, through the successive hydrogen proton insertion from outer membrane to inner membrane, it will convert into the water. Okay, then hydrogen concentration will be high from. This is a very important enzyme, ATP synthetase, and it is present in the plasma membrane of organism. And when H plus ion concentration is increasing, because in the outside the cell membrane, so what ATP synthetase will do, from outside to inside, it will exchange the proton. From outside to inside, when it will take the H plus, and the ADP molecule react with the phosphate molecule, and it will convert into the ATP molecule. So your ATP molecule will produce in the electron transport chain. Now, if you will see overall net ATP production, so in glycolysis we have 8 molecule of ATP production from the 1 molecule of glucose. Then in pyruvate dehydrogenase we have 6 molecule of ATP. Then citric acid cycle 24 molecules. So net 38 ATP molecule will be produced from the 1 molecule of the glucose. Okay. Now, ATP will form aerobic oxidation of glucose by eukaryotic cell. So prokaryotic cells, they are doing different type of energy production, and if we will talk about the eukaryotic cells, they are also producing different type. So if you see eukaryotic glycolytic lytic pathway, they are producing total 8 molecules. Okay, and 2 pyruvate which is coming from the glucose. These 2 pyruvate to acetyl coenzyme, they will produce 6 ATPs. Then TCA cycle, they will able to produce total 24 ATP molecules. So combine the 38 molecules through aerobic yield from glucose, they are producing in the eukaryotic. Anaerobic respiration. So in anaerobic respiration, the final electron acceptor is not oxygen. We already discussed the in aerobic reaction, final electron acceptor is oxygen. But in anaerobic respiration, final electron acceptor is NO3, nitrate, nitrite, N2O, sulfate, and carbonate, and occur in the string anaerobic and facultative anaerobes only. So it is a very important that these anaerobic respiration is happening on the anaerobic type of microorganism, not every type of microorganism. So some pseudomonas, bacillus, they will use this type of anaerobic respiration. And ATP generation varies from bacteria to bacteria and always less than aerobic respiration. So the ultimately the in anaerobic respiration, the number of ATP production is less than to aerobic respiration. It is not more than aerobic respiration. So it is always less than uh, compared to aerobic respiration. So alternative electron carrier are used in the electron transport chain. That's why these electron acceptor will be some other molecule, not oxygen. Okay. Then example of this anaerobic reaction is fermentation. So in the fermentation, anaerobic reaction is happening, and it don't require any oxygen molecule, because the ultimately the oxygen carrier molecule is not oxygen. So it don't require any oxygen. That's why without oxygen this reaction can able to complete. So inorganic electron acceptor is required, and they all absence in the electron transport chain. So terminal electron acceptor is always organic compound, not oxygenic compound. So oxygen is not required, and the organic compound terminal electron acceptors is there. Then end product depend on the type of microorganism. See, different type of microorganism, they are producing different type of end product. So we'll discuss how these different type of microorganisms they will produce. They will utilize pyruvate and then they will convert into different type of product. See, pyruvate in the Clostridium, they will utilize the reaction in the anaerobic condition. They will convert this pyruvate into the butyric acid. Then gluconobacter is there, which will convert pyruvate into the acetic acid. Okay. Then pyruvate in the protease, what they will do, they will convert into the carbon dioxide and hydrogen. Then in acetobacterium, they will also produce acetic acid. Then enterobacter, they will produce 2,3-butanediol, and propionibacteria, which will able to propionic acid, it will produce. So different bacteria, they are having different cycles, and enzymes they are utilizing to convert into pyruvate into um, different type of product. So mixed acid they are producing, and some are producing very specific type of acids. Okay. So here is the example where the industrial important production is happening using the anaerobic cycles. And see, through the glucose initially molecule, initially carbohydrate we are providing to react, and through glucose they will produce the convert into the pyruvate. So two molecule of pyruvate will produce from the glucose, and this pyruvate will be utilized by the anaerobic respiration. And through anaerobic respiration, lactic acid will be produced, lactobacillus streptococcus, and they will do this reaction. Then yeast, yeast can able to produce ethanol and carbon dioxide. Then E. coli can be able to produce acid and other product and carbon dioxide. Then Enterobacter, they will neutral and product they will produce, which is not acidic or not a basic. And then some bacteria like Propionibacterium, they will produce propionic and acetic acid. And this propionic acid and acetic acid we can use to produce cheese and paneer. Okay. Then Clostridium bacteria can able to produce pyruvate into the butyric acid, butanol, and acetone. Okay, and these butyric acid, butanol, and acetone we can use for industrial purpose. Then lactic acid fermentation also there, and lactic acid fermentation used to use the glycolytic pathway and directly reduce almost all their pyruvate to lactate with the enzyme lactate dehydrogenase. So lactobacillus species is there, and they are using this lactic acid fermentation. So lactic acid, pyruvate to lactate or lactic acid they will produce, and lactate dehydrogenase is utilized. Then hydrolactic fermenters also there to form substantial amount of product. And other than lactate, many product like lactate, ethanol, and carbon dioxide is produced. Then catabolism of other substrate. See, protein is there, carbohydrate and fat molecules is there, and this protein is made up of amino acid. Carbohydrate is made up of monosaccharide. So monosaccharide is a main component of carbohydrate. Then fat is made up of mainly glycerol and fatty acid. So when amino acid is we are taking as a food material, they will deaminase, and through deamination they will able to convert into the pyruvate acetyl coenzyme A, and they will utilize into this citric acid cycle. And carbohydrate also, basic monosaccharide is a glucose, and it will also use into the glycolytic pathway, and it will convert into the pyruvate, and then it will go into this Krebs cycle, and through Krebs cycle it will go into the electron transport chain, and then it will produce finally ATPs. So total 38 ATPs will be produced by this aerobic respiration of the glucose. So fats can able to utilize convert into the glycerol, and glycerol can be utilized into the G3Bs of the glycolytic pathway. Then fatty acid also there. Fatty acid do the beta oxidation, and through beta oxidation they will able to produce acetyl coenzyme A. Then anabolism also there, and these basic molecules like inorganic molecules, monomers, macromolecules, or supermolecular uh, molecules also there. They are all the basically part of the cytoplasm. See, inorganic molecule is there: carbon dioxide, NH3, H2O, PO4, magnesium, calcium. These inorganic molecule is the basic molecules, and through basic molecules, nucleotide, amino acid, and sugars they are forming, and ultimately nucleic acid, protein, polysaccharide is forming, and combinedly they will form the membrane and various type of cellular uh, organelles and enzyme complexes, and complex cellular organelles like nucleus, mitochondria, ribosome, flagella, chloroplast, mitochondria is there. And combinedly, these cellular organelles is the part of the bacteria, algae, and all of. So these are all the some important references there, like microbiology by Pelzer. If you want to study further in advance topics, you can get detail of these topics of the microbiology Pelzer, microbiology by the Tortora, and Rock also there, and Prescott microbiology is there. Here is the some important question. First question is, which of the following is an organism that obtained its energy from the transfer of electron originating from chemical compounds and its carbon from an inorganic source? So you have to select the correct answer from the given four option. Then in lactic acid fermentation, the final electron acceptor would be, so four option is there: fructose, pyruvate, glucose, acetyl coenzyme. You have to select any right answer. Then a yeast of fungal cell produces how many net ATP molecules per molecules of glucose when completely oxidized? So 32, 34, 36, or 38. You have to select one correct answer. Then next question: before most molecules can enter the Krebs acid cycle, they must be converted to citric acid, auxiliary acetic acid, NADH, FADH, and acetyl coenzyme. So these four options is there, and you have to select one correct answer. Then how many oxygen molecules are required in the fermentation of one molecule of glucose to ethanol and carbon dioxide? So it may be 0, 1, 2, or 36. You have to select one or right answer. Then most bacterial fermentations yield how many net ATP molecules per molecules of glucose? A option is 1, B is 2, C option is 4, D option is 6. You have to select one right answer. Then which one of the following is produced in the greatest number during one term of the Krebs cycle? A option is NADH, B is acetyl coenzyme, then C is FADH2, then D is ATP. You have to select one right answer. Then aerobic respiration differ from anaerobic respiration in which of the following respects? So A option is anaerobic respiration is glycolysis. B is aerobic respiration requires the electron transport chain. C is the final electron acceptor are different. Then D is aerobic respiration produces less ATPs. The next question: in anaerobic respiration, the final electron acceptor can be A. It is oxygen. B is a nitrogen. C option is pyruvate. D is a acetyl coenzyme. So you have to select any one right answer. Then next question: which one of the following is not produced in many of the steps of glycolysis? So A option is NADH plus, B option is NADH, C option is ATP, and D option is ATP. So you have to select a right answer. So thank you with us. We'll see you in the next class.